What happens to sperm at high temperature? The egg and sperm, which fuse to form a zygote, make up an organism's germline. The germline can be temperature sensitive, meaning the egg and sperm function optimally at specific temperatures. When temperature strays from this optimum, germline function drops drastically. In organisms such as cows, pigs, humans, fruit flies, and worms, the sperm is known to be temperature sensitive. Our lab uses C. elegans as a model organism to study germline temperature sensitivity. These worms are ectotherms whose body temperature is controlled by the outside temperature. C. elegans are either hermaphrodites, which produce both sperm and oocytes, or males, which produce only sperm. For my experiments, I will be focusing on sperm produced by the males. Once sperm are produced, they are called spermatids. These immature spermatids cannot move. Spermatids undergo a process called spermiogenesis, where they activate and mature into movable spermatozoa capable of fertilization. Some worm strains, such as JU1171, are affected less by temperature and are thermal tolerant. Other worm strains, such as N2 and LKC34, are affected more by temperature and are thermal sensitive. C. elegans have the highest fertility around 68 degrees Fahrenheit or 20 degrees Celsius. At higher temperatures, around 80 degrees Fahrenheit or 27 degrees Celsius, fertility decreases to practically zero. When worms are moved back down to low temperature, fertility does not recover. We believe there is a problem with sperm function at high temperature that yields low fertility. We are asking questions such as, is there a difference in size, structure, or activation of sperm, or maybe a difference in mating ability between strains that are more fertile at high temperature compared with strains that are less fertile at high temperature. To look at sperm activation more closely, I will be dissecting out spermatids from males and activating them in vitro with chemicals such as pronase E, zinc chloride, monensin, DIDS, and TEA. I will be using antibody staining to compare the structure between spermatids and spermatozoa at both low and high temperature. Finally, I will be looking at male behavior at high temperature to see if they are capable of mating with a hermaphrodite. By the end of my project, I hope to have gathered enough evidence to suggest a plausible explanation as to why sperm do not function properly at high temperature.